With our third presentation of the afternoon by Bridgette Tonnsen. And the topic of her talk is From Cell Cultures to Community Cultures, Brining Precision Health to Autism.
Thank you. Hi everyone, my name is Bridgette Tonnsen. I'm an assistant professor of psychological sciences. I'm joined today by many members of our big team, if you want to wave, as well as my co-leaders, Dr. Mandy Rispoli, from my co-director of the Purdue Autism Research Center, and Dr. Ann Serino, professor of biomedical engineering and psychology. So let's begin today in the autism community, where you'll hear a familiar phrase: if you know one person with autism, you know one person with autism.
If you know a person with autism,
you know a person with autism.
This phrase is commonly shared by parents, self-advocates, friends, researchers, and community partners. We know that autism is a diverse spectrum, and each individual affected by autism represents a unique set of strengths and needs. Yet the current science that guides autism diagnosis and treatment tends to overlook the individual. Our fields have worked in silos and have largely ignored the intersecting, multifaceted factors that make each person with autism unique. Purdue University and Discovery Park are ideally positioned to radically shift this narrative. Our big idea is to establish Purdue as the world leader in personalized health for autism. Our team of experts have a strong track record of conducting high-impact, externally funded, and interdisciplinary research, with expertise ranging from cell cultures to community cultures. With support from Discovery Park, we will have the leadership, scientific ecosystem, community support, and infrastructure necessary to address our big idea head-on. Now is the time for Purdue University to emerge as a global leader in precision health for autism. Because as the saying goes,
did you know friend with autism? Did you know friend with autism?
So autism is a neurodevelopmental disorder that affects 1 in 59 US children and carries a public health cost of $256 billion annually. Autism, as you could see from the video, is a diverse spectrum. There are individuals with autism who work and study right here at Purdue, but other individuals with autism have significant unmet needs. For example, out of every 10 individuals who is diagnosed with autism, 3 will not develop the ability to speak. About half will not be able to hold a job independently in the community when they're in their 20s. About 3 will engage in self-injurious behavior or have intellectual disability. And about half are at risk for elopement or wandering, which is actually the leading cause of death among individuals with autism. Importantly, we only know the cause of autism in about 20% of cases, meaning the vast majority of the time when somebody is diagnosed with autism, we don't know where their symptoms are coming from. What we do know is autism causes likely emerge through a complex intersection of many different factors, including genetics, exposures, environmental variables, and these are packaged in a way that will be different according to somebody's sociocultural context. Yet the way our field studies autism is often in scientific silos. In other words, with the current status quo of how we conduct autism research, it isn't possible to look at how these different factors intersect within an individual. Our big idea is to change this narrative. So here at Purdue, our big idea is to establish Purdue and Discovery Park as world leaders in precision health for autism through POCKET. So POCKET, or the Purdue Omnityping Kit for Individualized Treatment, is a novel assay that parents can complete in their home to assess their child's risk for autism. This work builds on a lot of existing research here at Purdue that I'll share with you today. But in particular, it builds on my own research program as well. So I've been studying early risk factors for autism for over a decade, and I obtained NIH funding to develop PandaBox. This is a precision health approach to assessing clinical symptoms of autism in the family home. So our team is currently mailing this telehealth-based assessment of clinical autism risks to families across the country. And our goal with POCKET is to extend this to environmental and other types of exposures as well. Our team is composed of leaders across campus, representing basic science and genetics, engineering, public health, speech and psychology, education, human development, the liberal arts, and big data and statistics. We represent 8 Purdue colleges, 5 Discovery Park institutes, and importantly, our team has already obtained over 35 relevant active grants that will lay the foundation for this work. Our approach is to bring POCKET to the limelight through 4 main initiatives. First, we will mobilize and expand the Purdue Omnityping ecosystem, including by developing a patient registry through cost share with PIN and in conjunction with IU Health, particularly Dr. McNally-Keene, who's joined us here today. We will also optimize POCKET for data collection, including by conducting surveys and historic analyses that will map the sociocultural landscape that will affect how we design and implement Pocket in diverse communities. We will then examine the uptake, usability, and feasibility of Pocket in 3 diverse samples, including my genetic risk cohorts. So these are children with rare syndromes that are highly comorbid with autism who are already engaged in research projects here at Purdue. An environmental risk cohort by Dr. Ellen Wells. These are children who were born or raised on a Superfund site in Lake County, Indiana, and have been exposed to high levels of arsenic and lead, which are known to be associated with autism risk. And then a clinical risk cohort obtained from patients in our community who are being referred because they're showing behavioral signs of autism. Across these cohorts, we will leverage the data we collect to advance discovery of treatment targets and novel omnityping algorithms. For example, we will develop individualized telehealth-based treatments that leverage our Pocket data. With support from PIN, we will develop the cell engineering core, we will develop novel Pocket assays and explore potential pharmacological targets. And we will bring these rich data sets together to explore novel omnityping algorithms that can truly link an individual's personalized diagnostic information with their treatment protocol. The path to funding for this project is already very clear. Our team has a strong track record of building extramural research programs. Our goal, though, is to submit at least 4 major grants in year 2 that will cross teams that are not already funded to work together. These are some of the grants that we think we'd be very competitive for. We will also send our team leaders to meet with NIH or NSF staff annually. We will participate in Morgan Center initiatives to secure industry funding. And we will submit an NIH Center grant within 6 months of completing the Big Award Challenge, which will catapult our team forward as a leader in the field. I've already explained to you that we have the perfect team to do this work here at Purdue. And with support from Discovery Park, we have unique potential to develop this novel assay system, Pocket, to link a patient's clinical data, their exposure data, their environmental data, to their own personalized treatment program, including both behavioral and pharmacological approaches that we can develop right here at Purdue. Importantly, this funding will help us also establish this long-term ecosystem where we can link our patient data directly to drug discovery by leveraging the iPSC cores and actually pulling data from our biospecimens that can help us generate new pathways to autism treatment. Now is the time for Purdue to emerge as a global leader in precision health for autism, because as the saying goes, if you know one person with autism, you know one person with autism. Thank you.